The red color is showing the low p-value. It means those bubble which are showing with the red color, they are showing more confidence on the results. It means we have to select that bubble which is closer to the one. Welcome to another video tutorial. In this video, I will tell you how we can interpret results of KEGG pathway analysis. So before we go into the detail of KEGG bubble charts, let's have a quick overview of KEGG pathway analysis. So as we know, KEGG stands for Kyoto Encyclopedia of Genes and Genomes. So then what would be the KEGG analysis? So this would be the analysis of network of gene products, which are usually proteins. Actually, the KEGG database is a reference knowledge base for deciphering genomes, and this initiative was led by Japan in 1995. Actually, the KEGG is an integrated database resource consisting of more than 15 main databases, and all of the information in the KEGG database is divided into three major system informations, genomic information, and chemical information. And all the KEGG databases can be divided into different components like KEGG databases, KEGG organisms, KEGG pathway maps, KEGG modules, and KEGG orthology. But out of all these components, we have mostly come across with these KEGG pathway maps. Here you can see the KEGG databases and the KEGG object identifiers, in which all the system information, genomic information, and chemical information is brought about through different databases. And here you can see there are the different prefixes, and one example is given against each information system. So here you can see the system information is met through by different databases like KEGG pathway, KEGG bright, KEGG module, KEGG disease, and KEGG drugs. Here you can see the KEGG pathway. Here is one example. You can see HSA04930. Actually, the each organism in the KEGG system is abbreviated to a three letters, like HSA stand for Homo sapiens, that is the scientific name of human being. Similarly, you can see the KEGG bright is mentioned with the help of BR, JP, KO, that is KEGG orthology. Similarly, KEGG modules are mentioned with the help of M, and the KEGG disease are abbreviated to H, and the KEGG drugs are abbreviated to D. Similarly, you can see the genomic information, in which the KEGG environment is mentioned with E, and KEGG orthology with K, and KEGG genome with T. And similarly, the chemical information like the KEGG genes, KEGG compounds, KEGG glycans, KEGG reactions, KEGG repair, KEGG R class, and KEGG R enzyme are mentioned with the different prefixes like compounds for C, and G for glycans, and R for reactions. And similarly, RP stand for reactant pairs, and RC for reaction class. Here is another important class, that is actually the enzymes nomenclature, that are mentioned with EC number. You have often seen this EC number along with different enzyme. So you can search these EC number in the KEGG database website. You can see each enzyme is tagged with a unique EC number that can search through the KEGG website, and it has a whole pathway. But today video is about the interpretation of results. So let's first go that how we can interpret the results of KEGG database. And here you can see this is actually KEGG bubble chart, which is showing the top 20 pathway of enrichment. So now let's try to understand that what are its component, which are mentioned on y-axis and x-axis. So among them, the first thing which we have to understand is actually that rich factor. So you can see actually the rich factor is the ratio of number of DAGs in a pathway to the number of all genes annotated in that pathway. So the next thing which we should, as there would be the more the richness factor, there would be the more degree of enrichment. You can see here there is 1 battalion biosynthesis pathway, it is showing the highest degree of enrichment. So the next thing is actually the p-value. You can see the lower the p-value, the greater the confidence of the result. You can see here the red color is showing the low p-value. It means those bubble which are showing with the red color, they are showing more confidence on the results. And the next thing is actually the size of gene number. You can see the bigger the size of this bubble, there would be the more number of genes. Now let's try to understand some examples of pathway enrichment and try to understand what are their meaning. So in this example, now let's try to take this first bubble. You can see here the first thing which can be interpreted with this bubble is actually the highest enrichment factor. You can see here this bubble is corresponding to highest enrichment factor 1. So what it mean? It mean this is that pathway which is showing the maximum number of DAGs out of all the annotated genes. So we can see other thing, you can correspond to the size. You can see this bubble has very small size. It means very few genes have been reported to this pathway. You can see this actually the battalion biosynthesis pathway is not often studied in different literature. So that is why you can see the small size mean very few genes have been reported to this pathway, out of which all showing the differential expression. So the next thing is mentioned with the help of this red color. You can see that red color corresponds to the low p-value. It means there is high confidence on the results and there is less chance of error. So now let's move to another example and try to understand some more bubbles. Here you can see here is another bubble which showing although big size but low enrichment factor. So what we can interpret from this bubble, it is showing the lowest rich factor, because you can see here this was showing the highest rich factor, but it is showing because it is more close to the zero, so it is showing lowest rich factor. So what does this mean? The other thing is its size. You can see the size is relatively biggest compared to the other bubbles. You can correspond to this size with this palette. So almost there are 30 number of genes belongs to this pathway that are actually the plant pathogen interaction pathway. You can see the what would be the rich factor. You can see here almost the rich factor is close to the zero. Let's suppose the rich factor is 2. So how the rich factor would be 2? Because we know that there would be at least 150 genes annotated in the literature which belong to this pathway, then the richness factor would be 2, because we usually know that richness factor is the ratio of number of DAGs divided by all the genes annotated in that pathway. Because we know the size is showing the 30 genes, so if we divide 30 with 150, then the richness factor would be 0.2. So it means there are many genes which have been reported in this pathway, and out of all those genes there are at least 30 genes which are showing differential expression in your phenotype belong to this pathway. So what else it is showing? It is showing, you can see it is showing the low p-value. Low p-value mean less chance of error. Now let's move to another example. Here now we will try to find out that what are the different most studied pathway in the literature. So what do you think? What is the most studied pathway in the literature? We usually know that if would be the low enrichment factor, it means there would be more annotated genes belong to that pathway. So what would be the most studied pathway in this? So you can see this bubble is showing like plant hormone signal transduction. This bubble is showing the low enrichment factor. You can see this bubble is even more closer to the zero, but you can see its size is small. So we will find the biggest size. So this is the most studied pathway in the literature. Why? Because it is showing the lowest rich factor. It means the highest number of annotated genes. So now we will try to find what are the top 3 DEGs showing pathway. So what would be the top 3 DEGs showing pathway? So it would be these three. You can see the biggest size belongs to plant hormone signal transduction, photosynthesis, and phenylpropanide biosynthesis. And what are the top two highly enriched pathways? We know that enrichment is closely related to the rich factor. You can see the higher the rich factor, the higher will be the enrichment pathways. So top two highly enriched pathway would be more closer to one. You can see the more closer to one is this and this. So these would be the top two highly enrichment factor. So far we have understood that how to interpret the richness factor, its size and the p-value. Now let's try to move that how to select our representative pathway in the KEGG pathway analysis. You can try to focus this bubble chart and try to find your representative pathway selection. So here I will tell you that what are the different attributes which we should keep an eye while selecting our representative pathway. So what do you think, which is our representative pathway? So according to me, the representative pathway in this bubble chart is actually the plant hormone and signal transduction. Why? Because this is the most biggest size bubble. Yeah, you can see among all of them, this is the most biggest size. You can see here the size is showing the DG number. So it means you also need to select the most biggest size bubble and which is with highest richness factor mean closer to 1. Let's suppose there are two bubble which are one is here and one is here. You can see if there is one here, it means which is closer to the one. It means we have to select that bubble which is closer to the one. You can see this bubble and this bubble is actually of the same size, but we are selecting this one. Why? Because it is more closer to the one. So it means our representative pathway is the most biggest size bubble which is closer to the one and which is showing the low p value. It means low FDR value. Like if there is so there are two bubbles which are on the same spots, but one is with the green color and one is with the red color. So which we will select after red color. So hopefully now you can select your representative pathway through the bubble charts. And here once you have selected your pathway, at the end you would see that there are files and which are tagged with different pathway ID, like here you can see Alzheimer disease pathway map that stands for Homo sapiens, and this pathway has a unique ID. You can see here in this transcriptome analysis, you can see here there are the different boxes which are mentioned in the green color. Actually the green color is showing the annotated identifier nodes. I mean all the genes which have been already annotated through literature belong to this pathway. So what does this pink mean? Pink mean the gene causing disease. Like here you can see these are the gene which are causing disease. And what are the blue color mean? Blue color mean these are actually the gene which are meant for drug targets. Hopefully now you can understand that what is KEGG pathway and how we can interpret and how we can select our representative pathway. So here are some frequently asked questions which are asked by the students. So the first question, which pathway should be selected for the RT-qPCR analysis validation? So my answer would be select the genes. Usually these genes are provided in the Excel list, FC value greater than 2 and P value less than 0.05. From that list, you can try to select that pathway, and that pathway has many genes. And from top 3 representative pathway, you can select your genes for final QRT-PCR analysis, or try to read literature and find which pathway genes are reported in article which are relating to your best phenotype. Like, I mean, it depends on your experiment that which type of experiment you are performing and which phenotype is giving the best explanation of your pathway. Here is one example, like if you are studying drought stress and there is pathway like cell wall related genes changes. Like if you are studying drought stress and literature says that whenever there is a drought stress, cell wall related properties of a cell changes. So you can try to find those genes which are related to the cell wall morphology. And in example 2, you can see if your experiment is related to salinity stress, we know that when there is salinity stress, there is actually the high quantity of osmoprotectant and antioxidants are released by the plants as a defensive mechanism, like proline. So you can try to find out some genes which are relevant to proline, osmoprotectant, and antioxidant. So I mean, the selection of RT-qPCR is based on actually the two criterias, among which the first one is the top three pathway, which I have just previously mentioned how we can select our representative pathway. The second is that your phenotype, how your the second is how your phenotype is being justified by different molecular mechanism. So among the list, the second question, I have many figures of KEGG pathway. Which should I select for the final article? So, well, it depends on the physiological data of treatments. If your all treatments, let's suppose you have four treatments, like let's suppose T1 is CK, and you are comparing at a time only two treatments, T2 with CK, similarly T3 with CK, similarly T4 with CK, and you find that all of them are significantly different, then you have to give the KEGG pathway up. Then you have to provide the KEGG pathway bubble charts of all the treatments. But if you think that there is no significant difference, then there is no need to provide the each bubble chart. So hopefully this video would be helpful in understanding the KEGG pathway analysis and KEGG bubble charts, and hopefully it will also help you in writing the results of KEGG pathway analysis. But if you are still facing any difficulty and still have another question, please let me know in the comment section. Thank you for watching. Bye bye. See, the lower the p-value, the greater the confidence of the result. You can see here